Hello, good evening, sir.
Hello. Yeah, good evening. Good evening. Yeah, we will be starting in just 2 to 3 minutes.
Yes, sir.
Okay. So, you have any questions from the previous class? So, hello and welcome everyone to today's week 4 live session on the course Experimental Biochemistry, which is being taught by Professor Shagota Dasgupta and Professor Shomode of IIT Kharagpur. I am Srijan Sen, and today I will be taking week 4 live question and answer session. So let us begin. So I would be firstly discussing with you the lecture slides in brief, and then I would be solving a few question and answers that will help you in solving the assignments and taking up the future exams. Okay. So I am sharing my slide with you all. Okay. I guess my slide is visible to you all. Means my screen is visible to you all.
Yes, sir. Yes, sir.
Okay, that is good. Okay, so let us begin. Okay, so in today's class, so for the previous two classes, what we discussed was mostly about buffers. Then we talked about amino acids and the structure of the amino acids, the titration of amino acids. In the third lecture, we spoke about the details of spectroscopy techniques, then Lambert-Beer's law, and we discussed a few things about the Jablonski diagram, about absorbance, transmittance, etc. Okay, so yes, so all of this is there in YouTube, and those who have not attended those sessions and all can definitely get back and listen to all of this, because and I guess these have already been shared with you people in by the NPTEL team. Okay. So I guess that part is done. So all of these lectures are also being recorded, so anytime you can get back and look at these lectures. Okay, so now in today's lecture, we will be mostly discussing about proteins, the structure of proteins and how to deal with proteins, what are proteins, and mostly the basics of proteins alongside protein folding and denaturation and renaturation studies of proteins. So firstly, in the introduction, when we talk about proteins, so what are proteins? So proteins are basically biopolymers which are made up of the 20 naturally occurring L-alpha amino acids linked by a peptide bond. So how do we get a peptide bond? So a peptide bond is that we have a NH2 group, we have a COOH group, NH2 group and a carboxylic group. So the water molecule is lost from, the hydrogen is lost from the NH2 group, and an OH group is lost from the carboxylic group, and then a bond is formed in between it, and that's a peptide bond. So proteins are actually so amino acids are basically the building blocks of proteins. So proteins, on the other hand, are biopolymers, which are made up of your naturally occurring. Maybe sometimes there are unnatural amino acids of L-alpha amino acids. So the polypeptide backbone is a repeating sequence of, so it's a repeating sequence of NCC backbone, and so this N comes from your amino or the N terminus in H2 group, then this is your C alpha and this is your carboxylic group, and again it gets repeated. So amino group, C alpha or the alpha carbon, the carboxylic carbon, then the amino group, C alpha plus 1 or the next alpha carbon, and then we have the next carboxylic group. So this is the repeating backbone sequence of the protein chain, or the protein backbone structure has a repeating sequence of this. And the side chain or the R group is not a part of the backbone or the peptide bond. So the R group is basically, so here where is the R group? So R group is attached over here. So we have the R group, which is not a part of the backbone, rather it is part of the side chain, and this carbon is known as the alpha carbon. Okay, and this is also another alpha carbon. So if it is alpha, this is alpha plus 1, and it continues like alpha plus 2, alpha plus 3. So this can be R1 and this is R2. So this is not a part of the backbone, rather it is part of the side chain. So this is a polypeptide chain because here we have seen that it is a biopolymer made up of 20 L-alpha amino acids made up of linked by peptide bonds. Okay, so we need to understand what is a polypeptide chain. So how is the peptide bond formed? So here we have 4 amino acids, and here you have methionine, aspartic acid, leucine and tyrosine. So what happens? So you have a carboxylic group over here, and here you have an amino group. So what happens over here is that water molecule is lost. So how is the water molecule lost? So water molecule is lost. So one oxygen is lost from here, and the other 2 hydrogens is lost from here. So water molecule that is lost is providing the energy for the formation of this peptide bond. So one oxygen from here and the 2 hydrogens from here is lost, and you get a peptide bond. In a similar manner, for us, the bond between aspartic acid and leucine and tyrosine, the water molecules are lost and you get a peptide bond. And this is your backbone. So in the previous slide I was showing NCC. So if you can see how is the polypeptide backbone being formed, see NC alpha C, NC alpha C, NC alpha C, and hereby it continues. And the side chains, that is the R group. So here you have this sulfur atom in over here. Here you have polar group. Here also you have a polar group. Then here you have an aliphatic chain or non-polar group. So these are all the parts of the side chains. But the major polypeptide backbone is formed by this nitrogen, alpha carbon and the carboxylic carbon atoms only. Okay. So this is a schematic diagram where this is the backbone and these are the polar side chains, these are the non-polar side chains. Okay. Now, what is the peptide bond? So peptide bond is formed by the dehydration reaction. So dehydration means loss of water molecule that provides the energy for the formation of a peptide bond. So OH and H. So this is lost and you get the peptide bond. Okay. So now we talked about the peptide bond. So we need to also talk about the characteristics of the peptide bond. So peptide bond, first of all, it is formed by the loss of one water molecule. Oh, okay. So if we have 4 amino acids, the number of bonds or the peptide bonds that will be formed is n-1. So for 4 amino acids, it will be n-1 number of peptide bonds. So if there are n amino acids, then the number of is n-1. Also, if it is now this is a linear peptide that is being formed. So this is n-1 for linear, but it will be n for your cyclic. So if it is a cyclic peptide, then it's a cyclic peptide, and this one is a linear peptide. For linear, the number of peptide bonds will be n-1. For cyclic, it will be n. Okay, now the characteristics of the peptide bond is that it is a covalent bond, and covalent bonds, as we all know, are strong bonds. And this has a partial double bond character. How do we know that? Because the distance is 1.32 angstrom, which is between your single bond. So your carbon nitrogen single bond has um, so if you think of carbon nitrogen single bond, it is 1.49 angstrom, whereas your carbon nitrogen double bond has a bond distance of 1.27, but this distance is between these two, so it is 1.32 angstrom. So it is between the single bond character as well as the double bond character. That's why it has a partial double bond character. So the peptide bond has a partial double bond character. At the same time, this bond is covalent, it is rigid, it is planar, and the prevailing trans conformation of the neighboring alpha carbon atoms. So the trans conformation is more relevant, and the side chains are free to rotate on either side of the peptide bond. So that is okay. Now, covalent bonds. So there are different types of bonds that can occur inside a protein molecule apart from your peptide bonds. So there are other bonds that also play roles that can occur between the side chains as well as the backbones. So in the backbone, the covalent bonds are your peptide bonds. Then we have the disulfide bonds that occurs between the two sulfur atoms. For non-covalent bonds, we have the hydrogen bonds, the ionic bonds, and the hydrophobic interactions. So hydrophobic interactions, how does it occur? So there are a few non-polar amino acids that forms the core of your protein, because it does not want to interact with the water molecules of the surrounding. So what it does, folds it folds to the inside of your protein molecule. It does not want to be exposed to the surroundings. So all of these non-polar side chain amino acids, what they do, they fold inside, and for holding the protein, the structure of the protein, these amino acids are actually uh, these amino acids actually interact among themselves, and just because these are non-polar aliphatic side chain wala amino acids, that's why, and also they are hydrophobic in nature, that's why we call them as the hydrophobic interactions, and these hydrophobic interactions are responsible for forming the core of the protein. Okay, so these are the typical bonds in your protein molecule. So we spoke about the peptide bonds. Now let us see what are these disulfide bonds. So okay, so these disulfide bonds are mostly formed between your cysteine molecules. So this is a singular cysteine and this is a plural cysteine. So what happens? So we know the structure of cysteine. It has a SH in the reduced form. In the reduced form, this cysteine stays in SH. Whereas when it is oxidized or in the presence of oxygen or an oxidizing agent, what it does, it forms a bond between these two cysteines, these two sulfur atoms. So there is a bond between these two sulfur atoms, and the bond between these two sulfur atoms is known as the disulfide bonds. So in the reduced state, it remains as a singular amino acid, whereas in the oxidized state, it remains as a disulfide bond. Therefore, we need to add some reducing agents to break these bonds. That can be your DTT, that is dithiatriol, and then you have the BME or beta mercaptoethanol. So between these two cysteine molecules, you have this disulfide bond. So okay, so one beautiful example of this disulfide bond is your insulin. So this is the amino acid sequence of your insulin, where the two chains are basically joined by two cysteine. Yes, so between two disulfide bonds, and also there is a disulfide bond. There is an intra chain disulfide bond, and these are interchain disulfide bond that holds the two chains together. So between two cysteine R groups of the polypeptide chains, this disulfide bond formation occurs. Hydrogen bond. So hydrogen bond, we know that this hydrogen bond is found between your positive partially positive hydrogen and your electronegative atom. So it can be between oxygen, can be between fluorine, nitrogen, or those group atoms, or means the group of atoms that falls in the category of nitrogen, hydrogen, and your halogens. So hydrogen can form bonds with that, provided these are electronegative in that particular molecule. Like if your oxygen is delta positive, then it cannot form a hydrogen bond. It has to form a hydrogen bond when it is electronegative than the hydrogen atom. So that is an hydrogen bond formation. So you have an electronegative oxygen, you have an electropositive hydrogen or a delta positive hydrogen, and the hydrogen bond is formed between this. Now ionic bond formation, also we call that as the salt bridge formation, because it's an electrostatic interaction between the charged amino acid, mostly your negatively charged amino acid with the positively charged amino acid, or vice versa. Like we have a lysine over here, and here we have an aspartic acid. So this is the anchor, and this is the positively charged amino acid, and aspartic acid is negatively charged. So there is an electrostatic interaction between these two amino acids or these two residues, and that forms the bond or an ionic bond. Like here you have the glutamate and here you have arginine, negatively charged, positively charged. So there is an ionic bond formation or an electrostatic interaction. So mostly these pH influences the strength of the ion bonds. In strong acids, it exists as NH3 plus and COOH, whereas in strong base, it exists as NH2 and COO minus. But there is an ionic bond formation between these two, say, these two groups. Okay. Now another interesting interaction is the hydrophobic interaction that is responsible mostly in the formation of the protein structure. Like over here you have this green portion. This green portion is the non-polar radicals and the blue portion is the polar radicals. So what you see over here is that the polar molecules are mostly staying on the outer portion or the surroundings to form hydrogen bonds with water, whereas the non-polar side chains are mostly involved in the formation of the core of the protein molecule, and this hydrophobic interaction between the aliphatic chains is responsible for holding the structure of that particular protein molecule. Okay, so this is an unfolded polypeptide, and in the folded conformation in the presence of water, the polar molecules or the polar side chain amino acid stays on the outer portion, and the non-polar side chain amino acid stays on the inner portion and forms the core of your protein molecule. So in summary, how many bonds we had studied about? So starting with, we have a hydrogen bond that forms with a water molecule or with a electronegative atom intermolecularly or intramolecularly, interchain or interchain that can occur. Then we have the hydrophobic interactions and the van der Waals interactions. Van der Waals interactions are very weak interactions. Followed by, we have the covalently strong bonded polypeptide or your peptide bonds that forms the polypeptide backbone. We have the cysteines forming the disulfide bridges. We have the salt bridge forming charged amino acids or the ionic amino acids, and it forms the ionic bond, and there is an electrostatic interaction, and these are the very important interactions for holding the structure of a particular protein in its tertiary state or a secondary state. Now, when we talk about the structure of proteins, there can be different structures of proteins. So, firstly, when we start with the bricks, that is your amino acids. So, firstly, only we do not jump to build up a house. So, we have to do it part wise. So, first comes the bricks. So, we arrange the bricks, we cement it, and then we arrange it. Then we go to giving it a particular structure. We then we make a few rooms. Then we give it. Then we paint. Then we make some other portions of the house. Then we go about putting all those coating and all, and then finally we give it a roof, and then we complete the protein structure. So the house structure. So in this same manner is also that occurs for proteins. So it starts with the primary structure, then we go to the secondary structure, the tertiary structure, and the quaternary structure. So the native protein structure or the primary structure of a protein is essential for its biological function. And the loss of structure of that particular protein also affects or hampers the functioning of that particular protein in the biological scenario. So we will start with the primary structure. So the primary structure is the basic structure of amino acids forming the backbone of the protein. So here you have the amino acids that are arranged by your peptide bonds. So you have the polypeptide backbone and you know the sequence of that particular protein. So that is the primary structure. So in the primary structure, you have an N-terminal amino acid, you have a peptide bonds and the C-terminal amino acids, and you know the sequence of that protein, of that particular protein. So your peptides can be dipeptide, tetrapeptide, tripeptides. Then you have oligopeptides that goes up to 20 amino acids. You have polypeptides from 20 to 50 amino acids, and then your proteins mostly are between uh, above 50 amino acids. And we know that a single change in the amino acid sequence in a polypeptide chain can change or completely destroy the protein function. Like for normal hemoglobin, we have, so this is the DNA sequence, this is the mRNA sequence, and here you have the glutamate. Okay, this is for a normal hemoglobin. Now when there is a single nucleotide that is mutated for CTC, it becomes CAC, so T becomes A. Okay, so thymine becomes adenine, and in similarly mRNA will form the transcript and this glutamate changes to valine. The moment there is a single amino acid change or a change in the nucleotide leads to a change in the amino acid sequence, it leads to a diseased condition. So what disease condition? A normal hemoglobin or a normal red blood cell becomes sickled red blood cell, and we call this as the sickle cell anemia. So previously it was all okay, it was normal, but only one amino acid is leading to a diseased condition. Similarly, when the structure of a protein molecule is lost, it causes loss of function. And loss of function is moreover okay, but what occurs mostly is that it leads to a diseased condition. Okay. So now secondary structure. So secondary structure is the arrangement of the regular arrangement in space of adjacent amino acid residues in a polypeptide chain. So secondary structures can be of three types. You have the alpha helix, the beta sheet, and the beta turn. So helix, sheets, and turns. So what is an alpha helix? So alpha helix is formed. So we can know that it's an helix. That is, it is a coiled structure. From the helix, it's a coiled structure. So this coiled structure is formed as a result of the hydrogen bonds between the carbonyl carbon and the amide hydrogen of the polypeptide chain. And it does not depend on the side chain radicals. So the R groups remains wherever it is. So the backbone is involved in the hydrogen bond formation. So how does it occur? The carbonyl carbon of each peptide bond is linked to the hydrogen bond with the amide hydrogen of the fourth amino acid towards the C terminus. So this is very important. So you have carbonyl carbon. This is your nth carbon. This is your nth amino acid, and then you have 1, 2. This is 1, 2, 3, and 4. So this is N plus 4. So this hydrogen bond is formed between the carbonyl carbon of your first amino acid and your between the carbonyl oxygen of your first amino acid and the amino hydrogen of your fourth amino acid. This is very important. Hydrogen bond with the amide hydrogen of the fourth amino acid towards the C terminus. So this is your N terminus and this is a C terminus. Okay, and the pitch of an alpha helix is 3.6 residues per turn. So the pitch of this amino acid is 3.6 residues per turn. Now what are the features of alpha helixes? So it is most common and it has a stable conformation. It has a spiral structure, that is the polypeptide bonds form the backbone of the spiral. R groups of the amino acids remains outwards of the spiral, and this is stabilized by hydrogen bonds. So hydrogen bonds are weak but collectively determine the stability of the alpha helix. Now the question is that now we have an alpha helix, but there are several amino acids that disrupt the alpha helices, or there are several situations that can lead to disruption of alpha helices. So for example, if we have a proline or glycine that is say prolines are very rigid amino acids, whereas glycine are very flexible, and these are mostly your helix breakers. So presence of proline or glycine can disrupt your alpha helix. Whereas if there is an equally charged radicals that is crowded around a particular place, so more than forming that hydrogen bond, there would be repulsion. Like if we have an equally charged radicals like lysine, arginine, histidine, or say glutamate alongside aspartic acid, that is actually a problem. Now the crowding of bulk R groups like leucine, isoleucine, tyrosine, that leads to spatial interference because it cannot form that coil or that coil-like structure. That helix cannot be formed because of the overcrowding of bulky R groups. And then amino acids with R groups which are capable of forming hydrogen bonds, that also sometimes forms intra, more than forming that intra helical, it forms the interhelical hydrogen bond that also sometimes disrupts the alpha helixes. And then the presence of certain chemicals like carbamide also disrupts this alpha helix formation. So we learned about the alpha helix and the features and how can they be disrupted. Next, coming to the second type of secondary structures, that is the beta pleated sheets. So these beta sheets can be of two types, the antiparallel beta sheets and the parallel beta sheets. So the antiparallel beta sheets are the polypeptide chains that runs in an opposite direction. So from here you can understand. So if you have the C terminus over here, this is the N. So here you have C and here you have N. But in the parallel beta sheet, you see N terminus alongside N terminus, C terminus alongside C terminus. So they run in the same direction with longer looping sections between them. So here you see this is the anti-parallel beta sheet and this is the parallel beta sheet, where these are with longer loop sections. So these loops or these longer loop regions are your beta turns. So these beta turns permits the change of direction of the peptide chain to get a folded structure. So the beta turn loops allow for the protein compaction. It releases the stress since the hydrophobic amino acids tend to be to the interior of the protein, while the hydrophilic residues want to be outside to the aqueous environment. Now, for these two beta sheets, for the antiparallel and the parallel, there is a hydrogen bond that is formed between this carbonyl oxygen and the amide hydrogen, and it occurs in both the cases. Okay, so so this is all about the secondary structure. We have the alpha helix, beta sheet, and the turns. Right. So next we would be talking about the tertiary structure. So this tertiary structure is a complete three-dimensional shape that is given to a protein, and we basically call that as the conformation of the protein. So it represents the spatial relationship of the different secondary structures to one another within a polypeptide chain, and how these secondary structures fold into a three-dimensional form of the protein. So in the primary structure, we only knew the sequence, that is the basic sequence of a particular protein. And these sequences fold from hydrogen bonds, from different bonds, to give us a secondary structure. Now these secondary structures join or add up to give us a tertiary structure that is actually responsible for the conformation of the particular protein. So the tertiary structure describes the relationship of different domains to one another within a protein. So what is a protein domain? So a protein domain is a basic structural unit within a protein molecule. Like you have the alpha beta alpha, the beta beta alpha, you have beta alpha beta, then you have the beta barrel structure, you have the there are different other the Greek key motif and all. So these are the different domains that actually exists. So that is a part of the protein that can fold into a stable structure independently. And different domains can possess different functions. So proteins can have one to many domains depending on the protein size. A polypeptide chain with 200 amino acids consists of two or more domains, and these domains are usually connected with the relatively flexible areas of the protein. So the tertiary structure is based on various types of interactions between the side chains of the peptide chain. So what are the interactions that are involved? So that is the hydrophilic interaction to water. Then we have the alpha helix that is formed, and there are intra hydrogen bonds because of which this alpha helix is forming. We have the hydrophobic interaction between the aromatic chains, then you have the aliphatic chains or the non-polar amino acids. You have other beta-pleated hydrogen bonds. There are disulfide bonds that can also be intra or inter. You have the salt bridges of the electrostatic interactions, alongside there are van der Waals interaction and other small or weak interactions that occurs, and all of these is responsible for the tertiary structure of a particular protein. So these are the stabilizing interactions of the tertiary structure. You have the hydrophobic interactions, that is the attractions between non-polar alkyl and aromatic groups form a non-polar center that is repelled by water. For example, all of these that are examples. Then you have the hydrophilic interactions. So hydrophilic interactions are attractions between polar or ionized R groups and water on the surface of the tertiary structure. You have salt bridges, that is ionic interactions between ionized R groups of acidic and basic amino acids. Hydrogen bond that occurs between polar side groups of amino acids. And finally, the disulfide bonds. Those are strong covalent bonds between sulfur atoms of two cysteine amino acids. So in the reduced state, it remains as SH group, and in an oxidized state, it forms a disulfide bond. Now coming to a complex structure of these amino acid that is also formed by the quaternary structure. So this quaternary structure can be of by monomeric proteins, that is built of single polypeptide chain. Also this can be formed by the oligomeric proteins, that is built of more than one polypeptide chains, called subunits or monomers. So quaternary structure describes the joining of two or more polypeptide subunits. These subunits have their own tertiary structure. The bonds are basically non-covalent interactions. These subunits can either function independently or can work cooperatively, and the dissociation of a subunit results in loss of function. So what happens sometimes, for example, you have one particular subunit that is joined by, say, non-covalent interactions, as it says. Now, so you have a particular subunit, but that does not actually function on its own. So what do you require? Maybe there is another similar kind of a subunit, say another subunit, the same protein having say 2 to 3 subunits. Okay, it forms an oligomer. Okay, this oligomer functions is more functional than the monomer. So you have a monomer, but it does not function well. Say there is another subunit, say 2 to 3 subunits appear, and these 2 to 3 subunits are responsible for the function of the protein. So, but when one of that subunit is lost, it stops functioning. So in that manner, the subunits actually can either function independently or can work in the presence of one another, that is cooperatively. So this is the structure of hemoglobin. So it consists of 4 subunits, 2 alpha chain and 2 beta chain of 2 different types, alpha and beta. And each subunit contains a heme group of oxygen binding, and the binding of oxygen to one heme facilitates the oxygen binding by the other subunits, and replacement of even one amino acid in the primary structure with another amino acid can hamper the function of that particular protein. So protein structure is hierarchical, that is primary structure is necessary for the sequence. The secondary structure is important for the local folding. The tertiary structure is involved in the long range folding. The quaternary structure is responsible for the multimeric organization, and then there are supramolecular or large scale assemblies that is also found. So we all know that structure determines function, and when structure determines function, this molecular structure is what we need to keep in mind, and what are the functions that proteins are basically involved in. So there are a huge number of functions, the mostly being the regulation or the signaling of different on-off modes that the proteins go through. There are signaling pathways, then you have the transport mechanism, enzymes proteins act as enzymes in different catalysis, then there are involved in different movements, and it is also responsible for the structure. So structure determines function. So what determines structure? So we have the energy and the kinetics. And how can we determine structure? For that we have experimental methods like NMR, X-ray, cryo-EM, and there are other computational tools for determining the structure. Like there is an AlphaFold that nowadays works very fine in determining the structure of a particular protein. Okay. So next we would be starting with protein folding. Okay, and so we have a few more slides to discuss. So let us take a few minutes of break, and then we can start with the protein folding and protein denaturation. Okay, so just a 5-10 minutes break. Okay, so let us start. So the amino acid sequence of a protein determines the tertiary structure of that particular protein. So this has been already shown by Affinson in Affinson's experiment, and although there are a few exceptions to all of these, like you have that all proteins can be denatured. Some proteins have multiple conformations. Then yes, then some proteins get folding help from chaperones, and the function of a protein is determined by its 3D fold. So it's important to see, can we predict 3D fold of a protein given its amino acid sequence. So the Leventhal paradox says that given a small protein 100 amino acid assumes 3 possible conformations of peptide bond. So 3 to the power 100, that is about 10 to the power 47 conformations. And in the fastest motions, 10 to 15 seconds per sampling. So it would take about 5 into 10 to the power 32 seconds. That is about 10 to the power 25 years. But it does not happen. So each protein folds quickly into a single stable native conformation, and that is the Leventhal paradox. So the overview of energy. So these are the different energies, like we have the hydrogen bonds between neutral groups, the peptide bonds. Then there are ionic interactions like attraction, repulsion, hydrophobic interactions, and van der Waals interactions. So these are the strength of the different bonds or the interactions that is responsible for holding the structure of the protein. And this is experimentally determined by the hydrophobicity levels. So the more positive this value is, the more hydrophobic is that amino acid, and the more negative that the value is, the more hydrophilic is that particular amino acid. And all of the combined effect of these values, depending on the sequence of the amino acid, tells us about the hydrophobicity or hydrophilicity of that particular protein. So what is the hydrophobic effect? So the conformation of a native cell, protein is only marginally stable. So conditions differ from cells to cell, therefore it causes protein structural changes, result in the loss of three-dimensional structure, and when there is a loss of three-dimensional structure, it is called denaturation. So in the denaturation state or the denatured state, the conformation of the protein need not be completely randomized. The number of physical and chemical agents can cause protein denaturation. Some of them includes heat, which has complex effects on many weak interactions. So on heating, it destabilizes the bonds and it affects the protein's conformation. However, the protein's conformation remains intact until there is an abrupt loss of structures over a relatively narrow temperature range. And the abrupt loss of structure suggests a cooperative process in which the loss of structure is one or more parts of the protein that destabilizes the structure of the other parts. Also, these proteins can be denatured in the extremes of pH, miscible organic solvents like alcohol, acetone, by certain solutes such as urea, guanidium hydrochloride, or by detergents. and none of these agents breaks the covalent bonds. Organic solvents, urea and detergents, it disrupts the hydrophobic interactions and that produce the core of that globular protein. On the other hand, urea and guanidinium hydrochloride disrupts the hydrogen bonds. So the extremes of pH that alters the net charge on the protein, it causes electrostatic disruption or electrostatic repulsion and the disruption of some hydrogen bonding. So the denatured structures resulting from these various treatments are not necessarily the same. And lastly, what happens is that the denaturation often results in aggregation and precipitation of the unfolded protein. The protein precipitate that is seen, um, you often see that after boiling an egg white is also a certain example. Now the question is, how do you renature a protein? But before that, what is more important from these two slides is that the first one being is that on heating, what happens is that a protein's conformation generally remains intact unless there is loss of structure. And the classical agent for denaturation is heat. Whereas there can be other things like pH, there are miscible organic solvents like you have alcohol and acetone, you can have urea and guanidinium hydrochloride, or by detergents. So urea and detergents disrupts the hydrophobic interaction. Another very important thing is that none of these agents breaks the covalent bonds. Then urea and guanidinium disrupts the hydrogen bonds, and extremes of pH alter the net charge on the protein, causing electrostatic repulsion and the disruption of some hydrogen bonding. Okay. Now, how will you renature proteins? So, previously studies have shown by that renaturation of the enzyme is provided by an experimental proof that the tertiary structure of the protein is determined by its primary structure. So what happened in that is that a purified ribonuclease was denatured and it was exposed to a concentrated solution of urea and reducing agent mercaptoethanol. Now this unfolded the protein and reduced all the disulfide crosslinks to cysteine residues. Now when urea and mercaptoethanol were removed, the randomly coiled denatured ribonuclease spontaneously folded to its catalytic active state, and even the protein's 4 unique disulfide bonds reformed correctly. And this was the first evidence that the amino acid sequence of the polypeptide chain contains information that is required for the folding into the native or the three-dimensional structure. So what happens? So it was a native state and it was catalytically active, and you add urea and mercaptoethanol, so it reduces the disulfide bonds and it unfolds the protein. And now on the removal of urea and mercaptoethanol, it folds into its native structure. Whereas it was also seen that only when mercaptoethanol was removed and it was made to fold in the presence of urea, it did not fold back into its native structure. Rather, there was a very random disulfide bond formation and it did not give a very stable structure. So this folding is a very slow process, as well as the proper bond formation is very necessary. So a harsh chemical or harsh condition does not allow it to fold. Now, thermodynamics of protein folding is that thermodynamically the protein folding process can be viewed as a kind of free energy funnel. So this is an inverted funnel. The unfolded states remains on the top of the funnel and are characterized by a high degree of conformational entropy and relatively high free energy. Now, as the folding proceeds, then it narrows down the funnel that reflects the decrease in the conformational space that must be searched as a protein approaches its native state. Now, some depressions along the sides of the free energy funnel represent semi-stable intermediates that can slow the folding process. And these funnels have a variety of shapes depending on the complexity of the folding pathway. The existence of the semi-stable intermediates that we can observe from these different peaks that suddenly rise in the middle. So it is supposed to be a very this is the best case scenario, but it does not occur. Sometimes there is an unstable state, and in this manner it leads to the most stable state. So these are the semi-stable states, and these funnels have a variety of shapes depending on the complexity of the folding pathway, the existing of the semi-stable intermediates, and the potential for particular intermediates to assemble into aggregates of misfolded proteins. Okay. So that is all about your protein folding and protein denaturation. Rather, protein denaturation and protein refolding. Okay. Now let us discuss a few practice problems. I guess these questions won't be very tough, but still let us discuss them. So the first question is, the end of a peptide chain has. So what does the end of a peptide chain have? Is it the alpha carbon, the carboxylic group, the carbonyl group, or the amine group? So if you remember from the structure that the beginning of the protein is known as the amino terminus and the end of the protein is known as the carboxyl terminus. So in the beginning you have the NH3 plus group and in the end you have the carboxylic group. So carboxylic group B is the correct answer. Now the residues that tend to form the interior core of the protein. So they form the interior core. What does that indicate? It indicates that these core is basically formed by the hydrophobic side chains that tend to cluster in the interior of the protein. Now the question is which has the maximum number of hydrophobic chain wala amino acids. So you have arginine, glutamate and leucine. So only leucine is hydrophobic. Tryptophan, phenylalanine and lysine. Here also only one. Phenylalanine, isoleucine and leucine. And valine, cysteine and aspartic acid. So if there is the presence of phenylalanine, tyrosine, tryptophan, leucine, isoleucine, valine, proline, glycine, alanine, methionine, so these would be the ones that forms the hydrophobic core. So here we have phenylalanine, isoleucine and leucine. So that has the maximum number of hydrophobic side chains and that forms the interior core of the protein. Now the question is, how is a peptide bond formed? Condensation reaction between amine and carboxylic group, elimination reaction of the carboxylic group, substitution reaction of the amine group by amide ligand and addition of H2O to the amine group. So the answer is very simple. It's the dehydration reaction or the condensation reaction between the amine and the carboxylic group. Okay. Now the question is the number of possible tetrapeptide sequences that are possible. So tetrapeptide sequence that are possible. So how do we do this? So how many tetrapeptide sequences can be possible? So there are 4 spaces. 1, 2, 3 and 4. So these are the 4 places. Okay. And for each place, how many is possible? So you have 20 into 20 into 20 into 20. Okay, so for each place you have 20 amino acids naturally occurring. So the product of all these 4 would be 16 and 4 zeros. So A, like 16,000. sorry, 1,60,000, because there are 20 possibilities in the first place, and that is all possible for all the 4 places. And just because it is a tetrapeptide means 20 into 20 into 20 into 20. Simple. The next one. In the peptide bond, the CN bond has. What does the CN bond have, or the carbon nitrogen bond have? The delocalized electron system, only double bond character, only single bond character, both single and double bond character. So for this, 2 options would be correct. It's a delocalized electron system and both the single and the double bond character exist, because the bond distance between that two is between the single and the double bond character. That is, it has a partial double bond character. Now the following pairs of residues that can form salt bridge in proteins. Salt bridge means ionic bonds or electrostatic interactions. So we have methionine, histidine, alanine, proline, aspartic acid, lysine, and arginine, glutamate. So here we also have 2 options that are correct. Positive and negative charges form your ionic bonds or salt bridges. So arginine, glutamate, and aspartic acid, lysine, both the options are correct, because negative, positive, positive, negative. Okay. The another one, the phi and the psi angles are formed around. Okay, so where are the phi and the psi angles? So let us get back to the previous structure. Okay, so if you see this, if you just see from this, let me try to draw. Where is the phi and the psi? So the backbone. So here you have N C alpha carbonyl carbon. So the bond between so these are bonds that is formed between 4 atoms. Okay, so between 4 atoms. So if you see, what are the 4 atoms? 1, 2, 3, and 4. So there is 1, 2, 3, 4. So here is 1. Okay. Next 1, 2, 3, and 4. So this is another. And then you have 1, 2, 3, and 4. So between N and C alpha. So one is between N and C alpha. One is between carbonyl carbon and your nitrogen, and the other one is between C alpha and carbonyl carbon. So these are the 3 bonds that can be possible. N, C alpha, C, N, then N, then C alpha, carbonyl carbon, N, C alpha. The other one is carbonyl carbon, N, C alpha, carbonyl carbon. So these are the 4 angles that are possible. So the angle between N and C alpha, that is N and C alpha, is your phi. The angle between C alpha and C is your psi. And the angle between C carbonyl carbon and amino group is your omega. So the 4 atoms forms the 3 angles, where you have nitrogen, alpha carbon, carbonyl carbon, and nitrogen, that is phi. Alpha carbon, carbonyl carbon, nitrogen, and C alpha, that is omega, and carbonyl carbon, nitrogen, C alpha, and carbonyl carbon, that is your phi. So now let us solve this question. What it says. Okay. Okay, so the phi and the psi angles are formed around. So the question is the phi and the psi angles are formed around. So let us see. NH, C alpha, C alpha, CO. So phi, so first is phi. So phi is formed around between N and C alpha. So this one, N and C alpha. And the psi is formed between C alpha and C carbonyl carbon. So option A is correct. Got it clear? Good. Now for alpha helix, the hydrogen bonds are formed between. So that question is very important. I said between the first carbonyl carbon and the NH of the I plus fourth. So this one and I plus fourth. So the correct option would be D. Carbonyl carbon of the ith amino acid and the NH of the I plus fourth amino acid. Okay. The next one. Which of the following is or are true for protein folding at the midpoint of the transition curve? So at the midpoint of the transition curve, what is true? That midpoint. It is already saying midpoint. So midpoint means the concentration of the native and the unfolded would be equal. Now when a polypeptide folds in an aqueous medium, the loss in entropy. So how is the loss in entropy compensated? So it is compensated. So when there is a polypeptide bond formation in aqueous medium, there is loss of water or release of free water molecules that is responsible for compensating the loss in entropy. There is a loss in entropy, so release of free water molecules that increases the entropy and it compensates it. Now for the protein unfolding reaction, if delta G greater than 0, which of the following statements is true? So delta G greater than 0, that means your reaction is not very feasible for a protein unfolding state. So the reaction is not feasible. So if you have a folded state to an unfolded state and delta G is 0, that means the equilibrium towards the unfolded state is not high, or the concentration of the folded state is much more. So what does that mean? Folded state is more stable because the reaction is not feasible to the unfolded state, or the reaction does not shift towards the unfolded state. And the folded state, what happens is that it is more stable, that's why it is pulling all the molecules towards the folded state, and that increases the delta G, making it more positive in a reaction where folded state, where the unfolding or the denaturation process is taking place. Now during the process of folding, which of the following does not occur? Now in the folding reaction, what happens? There is an increase in entropy, decrease in entropy, decrease in concentration of the native state, increase in concentration of the native state. So the thing that does not occur is that increase in entropy, free energy is correct, decrease in entropy is also correct, because that is what is compensated by the formation of the bonds and all, and there is an increase in the concentration of the native state, that is also correct. So decrease in the concentration of the native state does not occur. Now upon addition of denaturants with gradual increase in concentration to the protein solution, the absorbance of the protein increases as seen in UV spectrometer. Why does it occur? So what happens over here is that in the presence of denaturants, the protein unfolds, which exposes the amino acid residues towards the solvent, and greater the increase in concentration of the denaturant, more will be the amount of the protein that will unfold, resulting in the increase in the exposed tyrosine, tryptophan, phenylalanine residues, and that is responsible for increasing the absorbance in the UV spectrophotometer. So what happens? So there is a gradual increase in the aromatic side chains residues, and there is an unfolding of the protein. So when reactants are more abundant at equilibrium, which of the following is true? So we know that delta G is minus RTL and K, and that is the relation that exists between the Gibbs free energy and the equilibrium constant. So the equilibrium constant, if we try to arrange, so delta G, so where the reactants are more abundant. So reactants are more abundant means the delta G is positive. So delta G positive means that means K equilibrium is negative. So delta G positive and K negative. So B is the correct option. Very simple. It says the reactants. So reactants to products. Okay, so reactants more abundant means the reaction is not feasible. Reaction not feasible means delta G positive, and delta G equals to minus RTL and K. So delta G positive, delta K is negative. Very good. And the last question is, which of the following is not a protein denaturant? It's a very easy question. We have sodium dodecyl sulfate, guanidinium hydrochloride, urea, iodoacetic acid. So we all know that SDS is used in an SDS page for denaturing proteins. Guanidinium is also used for solubilizing the protein by denaturing that particular protein. Urea is a reducing agent that reduces the hydrogen bonds between the proteins, and it also opens up the protein. The only thing that is not a denaturant is an iodoacetic acid. So this is all from my portion about the assignments and the discussion of the session. If you have any questions, you can ask me.
Sir, yes. Hello, sir.
Yes.
Uh, well, when the temperature is given, enthalpy given, and entropy given, at that time, how to calculate the Gibbs free energy?
Sorry, I did not get you.
Um,
Okay,
react,
I got you. Okay, so for that we have an equation is delta G equals to delta H minus T delta S. Okay, so delta H is your enthalpy, delta S is your entropy, and T is temperature.
here is the one question is that calculate the Gibbs free energy for reaction at 27 degree where the change in entropy is 25 kilocalorie and the change in entropy is 50 calorie per Kelvin.
Yes, so that is good. So something you have got. So I said the formula, put all of that. So what you have? You have G equals to H minus T S. So all of this we know. So delta G is given. Your H is given as 50.
Ah,
50. 50
entropy
minus
calorie.
27. So 300 Kelvin.
Hmm.
300 Kelvin. And what is given as your S? Entropy?
Entropy 25 kilocalorie.
20.
25.
25 kilocalorie.
Hmm.
So 25,000 calorie. So now you calculate that.
The temperature is 27 degrees Celsius.
Yes, so 27 plus 273, that is 300.
Hmm.
So entropy is 25 kilocalorie or yeah. No, there is some mistake maybe.
20,
Enthalpy is 25,000.
25 kilocalorie.
Yes,
KCAL.
yes, yes. Yes. 25,000 is this one. This is 50. Right.
Ah,
So now that looks good. So you have what? So 25,000 minus uh, 300 into 50 is 15. 1, 2, 3. So it comes to 10,000. That is calorie. Okay, and that is in kilocalorie. Okay.
Sir.
Good. Any other question?
Hello, sir. Sir. Next one, another problem regarding to calculation. Calculate the gift free energy for a reaction where equilibrium constant is 2.5 raised to 10 raised to minus 3 at 298 K.
Okay, so that is what I said. G equals to, what is happening? G is equal to minus RT ln K. So this is 8.314 into, what is temperature? 298.
298 Kelvin.
And ln, what is given? K equilibrium 2.5.
2.5.
Yes, so this you have to calculate. I do not have a calculator. You put all
Uh,
these value in a calculator and you will get it.
thank you, sir.
Okay. Anything else?
Um, no, sir. Thank you.
Okay, good. So thank you. We will
Uh,
again meet in the next week.
yes, sir.
Okay, good.
Thank you.